Assalamu alaikum, welcome back to Ramzan Biology. Today our topic is about the life cycle of baterophage virus. In the previous lecture we talked about the structure of baterophage virus. And we studied that baterophage is a type of virus which suggests that bacteria mean bacteria and phage mean eating. So this is a type of virus which attack on bacteria and this eat a bacterial cell, destroy the bacterial cell. Now we talk about the life cycle of this baterophage virus. That's how this virus reproduce, how they increase its numbering. So let's talk about. There are two types of life cycle of baterophage virus. Baterophage virus undergoes two types of life cycle. The first one is the lytic cycle, the second one is the lysogenic cycle. So let's talk about the lytic cycle. Lytic cycle is also known as the master and slave relationship, in which the virus play the role of master while the bacteria play the role of slave. In this lytic cycle, a baterophage virus attacks on a bacterial cell and takes control of whole bacterial cell and work as a master, while the bacteria dependently perform the activity or perform the role of a slave under the control of baterophage virus. And the virus have the control, and is the virus one that it uses the bacteria for its benefits. So phage replicative cycle, which eventually cause the death of host cell. Phage mean the baterophage virus. It is the replicative cycle of the baterophage virus, in which the baterophage virus replicate itself, enter itself into the bacterial cell, then replicate itself inside the bacterial cell, and then eventually comes out of the bacterial cell and cause the death of the bacterial cell. So this is called lytic cycle. Okay, talking about a step. So the first step is the attachment or adsorption. Attachment means when something is attached to another thing. So here this is the bacterial cell, this one, and this is the viral cell. Okay, so this time the virus attach itself to the bacterial cell, and this attachment is done with the help of some protein. And so you can see here these protein are found on the surface cell wall of the bacterial cell, and these protein work as a receptor. And the protein fiber, tail fiber, you can see here these black fiber, these are called tail fiber. Then previous lecture we will talk about these. You should watch my previous lecture of baterophage virus. Now what is the function of the tail fiber? A tail fiber attaches the virus to another surface. So the virus attach itself with the help of this tail fiber and the bacterial cell with the help of these protein present on the surface of the bacterial cell. Now this virus cannot attach at this portion or this because there are no these protein. This protein are work as a receptor. Okay. The next step is the penetration. Penetration means going inside of something by pores. So this time, as we mentioned in the previous lecture, that there is a base plate of the bacteria of the viral cell, and this base plate have an enzyme called lysozyme enzyme. So this time the viral cell releases its lysozyme enzyme from its base plate and penetrate the wall of the bacteria. And on this way, when the wall of the bacteria is penetrated, so that time the viral cell contract with the help of sheath, flexible sheath, and the virus releases enter its genetic genome, meaning DNA, inside the bacterial cell. This is called penetration stage. Okay. The third one is the biosynthesis step. In this step what occur? So in this step we studied the DNA is enter inside the bacterial cell. So when this DNA genome of the virus enter inside the bacterial cell, so that time this DNA of the virus cell combine with the ribosome, with the help of ribosome. Using the ribosome of the bacterial cell, it degrade the genome of the bacterial cell. With the help of ribosome of bacteria, the DNA of the virus degrade the DNA of the bacterial cell. So and this way what occur, that after this biosynthesis occur, and you can see here that these blue, these are called the genome of the bacterial cell. They are degraded by the virus genome. So this time what occur, that inside the bacteria, when the virus enter, so that time the biosynthesis occur. Biosynthesis is the synthesis of biomolecule. So this time the viral cell replicate its head, its genome, its cell fibers. So in this way a lot of, from one viral cell, from one virus when enter inside the bacterial cell, a lot of other viruses are prepared and by the process of replication. Okay, so after this the cell assembly occurs. Cell assembly meaning again the assembly of the virus cell, again the assembly meaning again the combining of the machinery synthesis in the biosynthesis process. So here the head, tail and the tail fiber combine together and make a whole body of the virus cell. Then after that the fifth step, the lysis occur. Lysis mean releasing. So that time the virus again penetrate the wall of the bacterial cell and then comes out of the bacterial cell, and now they are prepared to attack on another bacterial cell and again run this lytic cycle. So the name lytic suggests that is a lysis, meaning the breakdown of the bacterial cell. Okay, now this was the simple process. Just I talk about the simple steps of the lytic cycle. If you want to take notes of this lytic cycle, so I show you a picture in which there is detail of each steps. Now let's talk about the lysogenic cycle. So lysogenic cycle is also known as the guest-host relationship. In this, virus play the role of a guest while the bacteria play the role of a host. So just like a guest do not damage to a host, so therefore a virus also do not damage the bacterial cell. Okay, so in this case virus live with the inside a bacterial cell with harmony with the bacteria. Okay, so let's talk about, this is a cycle in which page replicate its genome but do not destroy host cell. This is the replication of bacteriophage virus inside a bacterial cell but do not destroy the bacterial cell. It means the replication of the bacteriophage virus genome without destroying of the host bacterial cell. Okay, so let's talk about the steps. So just like the lytic cycle, the first step is the attachment. Just like in the lytic cycle we talked that the attachment of virus to the surface of a bacterial cell. The second step is same as the penetration. So just like the virus enter inside the bacterial cell, is called penetration. Okay, but the third step is different. It is called integration. In this step, just like suppose there is the virus, this is the virus and the virus enter its genome to this bacterial cell. So now the genome of the virus will be attached with the bacterial genome. Okay, and this is called then prophage. Prophage mean the attaching of the viral DNA attached to bacterial DNA. The attachment of virus DNA to the bacterial DNA, that is called prophage. But the real meaning of the prophage is the pro meaning before, while phage mean eating. So this is called the stage before eating. So now this genome of the virus is in the stage before the eating. Now it suggests, now we can find that this genome of the virus have the information to eat the bacterial cell, but it do not eat by now. So from here the lysogenic term is also found, that lyso mean the breakdown, genic mean the genome of the bacterial cell. So here in the lysogenic cycle there is the chance of damaging, destroying the bacterial genome, but the virus genome do not damage it. It have the information, it have the ability, but in this case, it live harmony with the bacterial genome. But after completing this cycle one time, if there is the chance, meaning some environmental condition damage or environmental condition changes, so it can start the lytic cycle back. Okay, now it is clear integration is the attachment of the viral genome with the bacterial genome. The fourth step is the binary replication. And binary replication, as we know that the replication of bacterial cell. So as the bacteria replicate itself and replicate its genome, so directly the viral genome will also be replicated. Okay, this step is called the pi-4 step is the division step, complete division, in which two daughter cells are produced, and each daughter cell have bacterial genome and viral genome. Okay, so from here to up to here, this is the lysogenic cycle. But after this, now here is the chance, if the environmental condition changes, meaning the condition, the atmosphere pressure or other temperature changes, or other changes in this lysogenic cycle changes, so it can enter to the lytic cycle. But if there is the change, if there is no changes, so it will again directly run this cycle again and again and again. हो लेकिन यहाँ पर हमारे पास lytic cycle फिर से शुरू होता है. मान लीजिए बन का हो रहा है. But here, after the division of bacterial cell into two daughter cell, if the environmental condition changes, so it can directly can lead to the starting of lytic cycle. But if the situation remain the same, so it will undergoes and run again this lysogenic cycle. Okay, so it can enter the lytic cycle, in which the virus, the genome, will lead to the synthesis of whole viral structure, and then will penetrate the wall of the bacterial cell and will go outside of the bacterial cell and will destroy the bacterial cell. This is the lytic cycle. So let us revise the lecture. First of all, lytic cycle is the master and slave relationship, in which the virus enter inside the bacterial cell and then penetrate the wall of the bacterial cell and then replicate itself and then comes out again. Okay, so this is the lytic cycle, in which virus damage the bacterial cell. But here the lysogenic cycle is a harmony cycle between the virus and the bacterial cell, and here only the genome of the virus attached with the bacterial genome, and when the bacterial genome replicates itself, so the genome of the virus also replicates, and eventually when two cells are formed, so both cells have the genome of virus cell. So this was about the life cycle of baterophage. These were the two cycle, lytic cycle and lysogenic cycle. So I hope this lecture will be helpful to you, and if you have any doubt or any question in this, so you can mention in the comment, and please subscribe and share my lecture by the next upcoming lecture, and also press the bell icon to receive update of my lectures.